hope you had a nice weekend. So the plan for today is I will give you first some definitions on these active transports that we were doing last time. Then I will show you some schematic diagrams to make you understand what they mean. Then we will set up some equations, try to solve some of them, and it will be the last time I talk about these active transport things, and then the next class I will do neurons. Okay, so something different to what you have seen so far. All right, so last time we started introducing this active transport through an example of a sodium potential pump. And I told you that if you want to move molecules against a gradient, then you need some energy, right? So this was the part where it was an active transport. It's not like diffusion that you have something that is passive, right? So for this one, you would have to expend some energy. So first, I want to define what is known as the Nernst potential. And this has to do with diffusion and electrical charges. So this is the potential that is obtained when all gates are open and there is a balance between diffusive flux and electric flux. So gates, basically what are they? They are protein line pores that allow the passage of specific molecules, right? So if you are unaware of what this means, I will write here they are protein line pores that allow the passage of specific molecules. So in what I will do today, you will see that you could have that a specific protein, for instance, has gates that are open and then they close, and then therefore you will have the transport of ions that is different at one time versus another. So we will be able to set up an equation for these different cases. So when I talk about diffusive flux, you can imagine if I have, for example, sodium inside and outside of the cell, right? You know that due to diffusion, these would try to move high concentration ions to low concentration, so you'll have diffusion. And when I am talking about electric flux, we might have that the sodium, which has a positive charge, is trying to move again through the membrane of the cell because you have a difference in charges, right? Positive versus negative. All right, so apart from this Nernst potential, we should also define what is known as the resting potential. So this is a bit different, and it's the difference now between the potential outside and far from the membrane of the cell, and that potential inside the cell. And basically, this one, the Nernst potential is talking about a specific ion, so it could be sodium, let's say, or potassium, right? But in this case, I'm talking about all ions together. So I will say here inside there, which may be different to the Nernst potential. Because, as we said, gates open and close and ions are moved under different ionic and concentration gradients. Okay, so this is kind of difficult to understand if you don't see a diagram. So let me explain what these different things are. So I will first introduce this Nernst potential schematically. So let's imagine that this line that I draw now is the cell membrane, okay? And again, I have the exterior and the interior of this cell. And I will tell you that in this specific case that we are drawing here, it's a membrane that is permeable to a specific ion X plus, but not to Y plus. So I have two different ions, you name them, and they have the same charge in this case, but one is X and the other is Y. X can easily pass through the cell membrane, but the Y cannot. And I will say that in this example, we have 13 X plus ions, and I have 17 Y plus. So this means that in total, I have how many positive charges? 30, right? Okay, now in the interior of the cell, now due to diffusion, I want to move from high to low. If I tell you I have X plus 7 of them, this is trying to move in this direction, right? And I will tell you that we have 23 Y plus here. So in the interior of the cell, we also have 30 positive charges, right? All right, now if I look at a different snapshot in time of this situation here, since we know that due to diffusion, we want to move from high concentration of this ion to low, and only X plus can move because of the permeability of the cell membrane, we will have, for instance, 11 X plus, and here I have two of them moved, so I'm going to get 9. And the Y didn't change at all because it's impermeable to it. Right? So this could be the scenario. So if I count now, the total plus is going to be 28. And in the interior, I'm going to have a build-up of 2, so it's going to be 32 instead. OK, so here I want to note that this is not going to keep going to 10, basically, due to diffusion on each side because of electrostatic potential pushing back. So it's basically an interplay between you trying to move from high concentrations of that ion to low concentrations, and the charges also trying to balance. When the two come in balance, it means that you are in equilibrium, and this is when the Nernst potential is reached. Okay, this is what it means. So here I'm going to make another note. There is going to be, in this specific case that I drew here, a charge imbalance. And this sets up an electric field which produces a force on the ions that opposes further diffusion of X plus. Okay, any questions so far? Yes?
Is this just a particular example?
Yes, this is a particular example. You might have to work on a different one in your problem set, but yes. I'm just saying in this case, I could set up also negative charges, and you would have to see when you are in equilibrium, basically between diffusive flux and electric flux. That's when you get the Nernst.
So if you had way more sort of Y plus on the interior, you could have greater difference?
Yes, yes. All right. So there is also the notion of a membrane potential. So I'm also going to do a schematic situation of that. And we're going to stress out what's the difference between the two. So the membrane potential is the following situation. Again, I'm going to do it through an example. So let's draw again the cell membrane. And I'm going to have X plus ions now and Y minus ions, OK? And I'm going to have the same amount of X plus and Y minus at the exterior and the interior, but a different number across the two sides. So I'm going to make here 5 X plus ions. And I'm going to also have 5 Y plus. So just as a note, I'm drawing this. And I'm going to have on the other side, again, an equal number of X and Y. Let's say we have 3, though. So it's different on the 2. So on this side, I have 3X plus and 3Y plus. Sorry, I will do something annoying. I'll just erase this and do a minus instead. Sorry that you already wrote it in the notebook. OK, so this is negative charges now. So this is, again, a membrane that is permeable to X plus, but not Y minus. OK, so what I'm drawing here, as I said, is just on each side you have equal and opposite charges, and on the other side, though, you have a different amount. So here we will have that X plus, again, diffuses through the membrane to balance the charges on both sides. So this happens very close to the cell membrane. The balancing happens close to the membrane. And in this case, you have that far from the membrane. You don't have any buildup of charges, so basically things are electroneutral. When I'm talking about liquids here, I'm talking about these salts that we said, right? Cells are made up of water with dissolved salts, we said. OK, and this, the potential difference that builds across the membrane as a result is the Nernst potential again. Okay, so these definitions are all nice, but we want to write down how we can actually model these cases. So I'm going to write down a decomposed form of the flux of these ions. That is basically the diffusion part and the electric flux as well. Okay, so it's going to involve a gradient of the concentrations for the diffusive part. And then we are going to have the gradient of phi, which is the electric potential, basically, to get the electric field. Okay, so what is actually the definition of this potential? What is this potential? So there are two parts of it, we said. There will be a flux of ions due to the concentration gradient. So the way I will write it is I will use J in general to write flux. This will be the first part of it. It's going to be a vector. It's minus d. And then I'm taking the gradient of C, where C is the concentration. D is the diffusivity. So maybe I will write these things down. So with a different color, this is going to be the ion diffusivity. And then instead, basically what this is the concentration. Okay. I will later simplify this to a one-dimensional thing so that we can work with it more easily. Okay. But for now, it's a vector. Okay, so this is the regular diffusive flux you have. But there will also be a flux of ions due to this potential difference we were talking about with the electric charges. So the way I will denote that is by J2. J2 is again a vector. So I'm going to write down some variables, and then I will describe what they mean. OK, so what are these U, Z, C, and so on? So I'm going to write them here. U corresponds to the ion motility. And it's the velocity if the ion is exposed to a constant electric field. Okay? Under a constant electric field. Z is something known as the valence of the ion. I'm not going to assume you know what this is. It's basically the charge. So if I give you Na plus, so sodium plus, it's just +1, okay? If I tell you you have O2 minus, the valence of that ion is 2 minus. So it's just the charge associated to that. So let's write it down so that you remember. The valence is just the charge of the ion. And as an example, I will give here sodium plus. This has valence +1. And if I give you O2 minus, this has valence -2. So basically, since we have here Z divided by Z absolute value is just +1 or -1, right? You're taking away the magnitude. So Z over Z absolute value is +-1, giving the ion charge. And I want to note here for you to know this. If I give you positive ions, so positive ions move down potential gradients. And if you have negative you can guess it, is going to move up. Okay, so this is still the concentration we had in the previous flux definition. And then phi, I will also define. Phi is the electric potential. So if I take now the gradient of phi, I'm going to get the electric field. Okay, so we can now we can combine the first flux, which is the diffusive one, together with this one from the electric charges to get the total flux for the system. So it's going to be just J. It's the sum of J1 with J2. And you have minus the gradient C minus U ZC d phi. I can also write it, let me just write it like this. So if I assume that this is now one-dimensional, so 1D, we're going to get that J is defined as minus D, dC dx, plus U Z C Z, and then I have d phi dx here. Okay? And when now, so basically we said that the Nernst potential, right, is when you have equilibrium between these two different fluxes, right? So if I have that J, the flux is 0, I can then solve for the Nernst potential and compute what this V is, correct? Okay, great. So before I do that, maybe someone could have asked me, how do you actually determine this D, right, the diffusivity constant? So this is something known as Einstein relation. So I'm going to write down the definition. If you want to learn more, you can read, you can search basically Einstein relation in diffusion or kinetic theory, and it will explain more, because it involves some constants that you determine empirically. So this is known as the Einstein relation. And it connects the diffusivity of an ion to the motility of that ion, so its movement. And it's basically going to involve temperatures and other things like the universal gas constant and so on of the ion. Okay, so its specific form of this D constant is going to be U RT divided by Z F. So U and Z are the things I have already defined, is the motility of the ion. And Z, we said, is related to the valence. And these RT and F are all constants. What the constants are basically, R is the universal gas constant. These are things that you know you can measure. T is the absolute temperature. And then finally, I have the F. F is known as the Faraday's constant. So basically, if you don't know what this is, I have written down the definition for you. This is going to be the total electric charge divided by the number of divided by the elementary charge carriers. So when you want to compute these things, the flux for a specific ion, these things, you will know what they are. OK, so now, as I said, we simplified it by writing it as a 1D equation. Now I want to compute this Nernst potential. So what we want to do is basically set J equals to 0, right? So the flux is 0. So in equilibrium, we have no flux. So this implies, maybe if I name this equation star, we have that d dc dx plus zc over z dphi dx is 0. And we can now integrate the flux, which is this equation basically. You do that from x is equal to 0 to x is equal to L. I will tell you what this is, to get. So when I say x is equal to 0 there and x is equal to L, this is basically saying from the interior to the exterior of the cell. And you will be given the boundary conditions to solve for this. OK, so when we get that, basically then you get, as we have already mentioned, the Nernst potential. I will denote by V. This is a definition. And it's going to be the result of integrating this thing. So I have it from the interior. So I'm going to denote the interior by an index I, and the exterior by E. So this difference in the electric potential is the Nernst potential. And it's going to be, after having introduced this D in here, I'm going to get a function that involves this universal gas constant and so on. So I'm going to get RT over ZF log of CE. So this is the concentration of the ion on the exterior of the cell membrane and the interior. OK? Any questions? OK. So E is for exterior. I is for, bless you, is for the interior. And then this thing that I wrote here is the Nernst potential across whenever I have to write down a name of someone, I think, oh my God, thank God that this thing is not my name. Imagine if the students had to learn to spell Mavroyiakoumou each time. It would be a nightmare. OK, sorry for the lame joke. OK, so now I will switch a bit. So now that we have defined the Nernst potential, I'm going to consider something related basically to circuits. Again, I'm not going to assume you know circuits, but we're going to talk about concepts like conductance, capacitance, current, voltage, all these things that you have in circuits, right? OK. So here we have the flow of ions across a membrane. We said it can cause a build-up of charge, right? This is what we have been talking about. So if you know a bit about circuits, you will know that basically when you have this build-up of charges, you will have something known as a capacitor. So this means that the membrane acts as a capacitor. So this means that now we can also compute, for instance, the voltage across the membrane. And it's going to be given as V, the voltage, is Q over C. So what are these things again? So Q is known as the, C is known as the capacitance. And this Q that I'm writing here is the charge difference. Okay, so this is the voltage across the membrane. We can also compute the current. This is going to be given as a derivative of the Q. So it's going to be minus dq dt. So depending on where you look, I think this can also be a plus. It's just convention. So we are going to be using minus for our class. So this means that basically the ionic current is going outside from the interior on the outside. OK, so now I will assume the capacitance is constant. OK, and then I'm going to basically say Q, the charge difference, is Vc, right? And then I'm going to take the derivative of that to get an equation that combines these two equations together. So we're going to have Cv is equal to Q from the voltage equation. And then C is a constant. So if I take the derivative of this, I'm just going to get C dv dt, right, is dq dt. But I know dq dt is minus I. So I have that C dv dt plus I is equal to 0. OK, this is basically the basis of what is known as electrophysiology. Bless you. OK. All right, so now we can also link the current of each species. When I am talking about species here, I'm referring to ions. To the voltage through the following equation. So I can write down IS. So here is just to denote that I'm talking about this specific species of ion. And I'm going to have GS V minus Vs. OK, so what is this GS that I wrote here? So GS is going to be the ion-specific conductance and what is this conductance now? So again, from circuits, if I use, do you know Ohm's law? Ohm's law is basically connecting voltage, current, and resistance. So if I want to relate them, is voltage is equal to current times resistance. This is these things known as Ohm's law. And we know from circuits, but if you don't know it, I'll tell you that conductance is basically the reciprocal of the resistance. So it's just 1 over R. And conductance is one over R. OK, so this is, by the way, what I wrote here is the Nernst potential. it was on the board before, but it's the thing we had, which is the difference between the exterior phi and the interior phi, right? The thing with the log. Okay, so if I want to compute the total current across the membrane, you can basically take the sum of these individual currents for different ions and get the total. So total current is I is just the sum over all the species of ions. If you want to think about this, it's just, since it's the reciprocal of the resistance, it just tells you how easily the membrane is letting charges through it, right? Okay, so now the question you should all ask me is, how do you actually find this GS, right? So how do we compute in our case? So this goes back to what I was mentioning at the beginning about gates, these pores that allow basically the passage of specific molecules. And we will write down some equations that you can basically compute the number of open or closed gates and so on. Okay, so the question becomes, what does this conductance look like? Okay, it's not going to be a constant. It's going to depend on the number or the fraction, if you will, of gates that are available. Again, I'm talking about specific species of ions here. So meaning which fraction of all gates are open. Okay, so to do this, I will now denote the number of open gates by n. So let's denote the fraction of open gates by n. In the notes that I will post, I sometimes refer to the gates as channels. If you see that, it's just the same thing. So then I'm going to show that GS is an GS max. And this is basically the conductivity when all gates are open. Okay, so when I'm talking about gates, you can think about it schematically like this. I have the cell membrane, and then there is a gate that can either be closed, or you can have, again, the cell membrane, and this gate is open, and it allows the passage of this specific species. Okay, so mathematically, I will go back to these reactions that we were doing at the beginning with enzymes. So if you remember, we were using the law of mass action, right, to write the differential equation. So let's assume now that on the left-hand side, I'm going to have the closed gates. And then here, I'm going to have open gates. And what you have is a reversible reaction that you can have gates that were originally closed, opening, opening, gates, closing, and so on. So I'm going to have here that the rate at which this happens is dependent on the voltage across the membrane. So I'm going to have here alpha. And then for the reversible, I'm going to have beta of V. Okay? All right, so if I want to write down a differential equation, I will use, as I said, the law of mass action. And remember this line here lets you know that I said the fraction of open gates by n, right? So basically, if I want to now keep track of what is happening to the fraction of open gates, I can write down the n dt. Does this increase or decrease? These are n, right? This is the number of open gates. So I'm decreasing the amount of open gates by beta, right? So I'm going to have minus beta of Vn. Does that make sense? And what am I going to have here? I'm gaining basically open gates. How can I denote the closed gates? If I have the total is 1 normalized, right? It's going to be 1 minus n is going to be the fraction of closed gates. So I'm going to have here plus alpha of beta 1 minus n. Is that clear? OK, so if you want to remember, this is basically the fraction of closed gates. Any questions? OK. All right, so sometimes you will also see this written in a slightly different way. So the way I will write it now is combine the n's together. So I'm going to get minus alpha plus beta, all of it multiplied by n, plus alpha of v. So I have the n dt is equal to alpha beta minus n, and then it's the sum of the two rates. I will divide through by this alpha plus beta, and I'm going to arrive at the following equation. I'm going to drop the v so that I don't just keep copying it all the time. It's going to be alpha plus beta minus n. And this I will be writing sometimes as this. Actually, this is, yeah, it's a different notation, n infinity. So where n infinity equals to alpha plus beta is the equilibrium value of n, of the number of open gates. And then tau is just 1 over alpha plus beta. And this corresponds essentially to the time scale for approach to the equilibrium n infinity. And basically, these values here are determined experimentally, so you don't have to worry about how you're computing them mathematically. OK, you can also have, this is the interesting case, you can also have multiple gates. So to keep track again of the equations, I'm going to use another notation. So I'm going to denote now by SI. This is going to be the density of states with I open gates. This means that if I write as 0, I just mean that you have 0 open gates, everything is closed. If I write, let's say we consider 2 gates. If I write as 2, they are all open, right? OK, so if I want to now write down an equation for the transition between gates, of different states, this is going to be governed by the following reaction. So I'm going to write it again in that form and then write the law of mass action. So I'm going to have S0, meaning everything is closed. This gets converted to state S1. And then S2 is all open, right? So this is 1 open, 1 closed. So the scenario we're considering is essentially the following. I have basically here, this is my cell membrane, and there is 2 gates that can either be open or closed. OK, so here I can have, I'm going to tell you why this has a factor of 2. This is going to be 2 alpha, this is alpha, and then this is 2 beta and beta. So let me write down a few things. This is the number of closed gates. And then basically the factor of 2. Let me get a different color. The factor of 2 is because we have basically 2 configurations. From going from 2 open to 1 open gate. I'm not going to write also for close, but you get it, right? Basically, if I have 2 open, this one can close or the other one can. OK, so once we have now set up this, you can also write down again, using the law of mass action, the equations for these states. So first, if I want to write, who can help me write down the equation for ds0 dt? I know you know. Yes, are you losing or gaining? Perfect, and then? Excellent. OK, so I'm going to write also the equation for 2. So for 2, you're losing the alpha one. Oh, no, so you're gaining from the alpha, so it's going to be plus, and then you're losing these 2 beta. So if you see these 2 equations, basically, if you want to write down the ds1 dt, is a combination of these 2 other things, so you don't have to write it down. We can infer S1 from the fact that the sum of these is 1. OK, so I have another 5 minutes. So basically, I will now use this, and I will write it back with the n that we had previously, which was denoting the fraction of open gates. So basically, this says we can reduce to one ordinary differential equation by finding that the solution is given by So here I'm going to write down the solution, and I'm going to just ask you to check that it's the solution, so it should just satisfy the equations. You don't have to actually try to solve it. So this is 2n, 1 minus n, and finally S2 is given by n squared. So how can I know that this is actually the solution? You just substitute back into the equations, and you see that it's true. Where now, if you do that thing I just told you, you can see that this thing is the equation we wrote previously. OK, so once we have this, basically this says that the proportion of open gates, which is this S2 state, the proportion of open gates is S2n squared. So the conductivity in this case, conductivity was the G I wrote previously, is given by G n squared, and then I have G as max. So it's the equation on that board over there. So remember, n was the fraction of open gates. In this case, the fraction of open gates is n squared. and then I have G as max. So you can actually generalize this to the case of n gates, where the conductivity in that case would be G is equal to n to the n, G as max. And then if you want to look at non-identical gates, you can also take a look at the typed lecture notes. I think it's on page 34, but we're not going to cover it in class. All right, that concludes these things about the ionic currents and so on. Next time, so tomorrow, we are going to look at neurons. So I'm going to look at a different model. OK, thanks for your attention.